Here we are showing the cycle of mitosis. With the cell cycle, the G1 phase begins with a small cell and then it grows. The S phase is a time when the DNA replication occurs and makes 2 copies. The G2 phase continues the growth of the cell, and the M phase is mitosis, which is what we are going to go over right now. I am showing you prophase, which is the first stage of mitosis. Inside of our nucleus, the DNA begins to coil and becomes these chromosomes. In this stage, the nucleolus actually also disappears, and the centrosomes start moving apart and creating these spindle fibers, these purple ones. Next stage is prometaphase. As I do this, the DNA is now coiled, and they're all each individual chromosomes. You create that little X, but again, it doesn't matter where they connect actually. It's just easier to do it right in the middle. They're kind of floating around there. And then the nuclear envelope has also broken apart and is gravitating towards those chromosomes. The centrosomes are at opposite ends, and the spindle fibers attach to the kinetochores of each of the chromosomes. So this is our prometaphase. Next up is metaphase, and this simply just means that the chromosomes move to the metaphase plate, and that means that they just move to the middle of the cell, like so. They move along these spindle fibers. So they are all on that metaphase plate. And that is our metaphase. Next phase is our anaphase. The chromosomes actually split at the centromere into 2 different daughter chromosomes. So they'll split right in that centromere, and they form 2 different daughter chromosomes. As this happens, the spindle fibers will actually shorten and pull the daughter chromosomes towards the opposite poles, which is why I am placing the daughter chromosomes closer to the centrosomes. They're still along the spindle fibers, but they are pulling them in opposite directions. And then the spindle fibers actually push at the cell, and it starts that elongation process to show that split of the cell. Next phase is our telophase, where the chromosomes begin to uncoil. And right now they are actually in the cleavage furrow. So the cells are not completely separated. They are still having that cleavage furrow in between. And then the nuclear membrane begins to reform. We still have our centrosomes. These are all separated. Our nuclear envelope comes back into play as each cell starts forming the nucleus again. And the spindle fibers deconstruct, which is why I took them out of the cell. Now there is one more phase, but it's not part of mitosis. It's the cytokinesis phase, and it occurs at the same time of telophase. But again, it is not part of mitosis.